Okay, well, welcome back. The moderator for this session is Dr. Robert Straka. I've already introduced him, so I'll ask Robert to the podium to introduce our final two speakers.
Thank you. Once again, welcome back. Today you've heard a lot of wonderful information about pharmacogenomics in general and a few specific areas as well. And all of this really matters when it comes to trying to implement things. So this next section, specifically called Implementation of Pharmacogenomics in the Real World Setting, is an area I think that everyone's primed for at this point in time. So we're very pleased to have two outstanding speakers that will briefly address what must be acknowledged is a critical area of active research and profound interest to all of us interested in advancing basically implementation of pharmacogenetic testing to optimize drug therapy. I'll start introducing Mark first, and then I'll wait until the next half of the session before I introduce our second speaker. It's my pleasure to introduce Mark Dunnenberger. Mark's currently a director of pharmacogenomics at the Center for Molecular Medicine, NorthShore University HealthSystem in Evanston, Illinois. As the director of genomics in the Center for Molecular Medicine, at that institution, Mark is a recognized leader, and throughout the country, a recognized leader in real-world experience implementation projects to improve the health care of patients and individuals that are subject to valued input from pharmacogenetic-based strategies. Mark's earned his PharmD from the University of Tennessee, Health Sciences Center, College of Pharmacy, after completing his PGY1 residency in Asheville, North Carolina. He moved on to do a PGY2 residency at St. Jude's Children's Hospital in Memphis, Tennessee. Mark has built one of the first of its kind pharmacogenomic practices by employing preemptive screening to enhance predictions of patients' responses to medications based on genetic makeup, precisely the topic we've been leading to at this, throughout this whole conference. His research and clinical interests include translating pharmacogenetics into clinical practice and developing informational technology solutions for utilizing genetic information to clinicians and patients in a practical manner. He also evaluates methods of educating healthcare professionals about pharmacogenomics, something we're all trying to do, and to enhance this whole area. A co-author of several CPIC guidelines, Mark brings his experience to our discussion today by way of his talk, Implementing Pharmacogenomic Testing in a Clinical Service, the NorthShore Experience. Thank you, Mark.
Thank you for that. Okay, can you guys hear me? We'll stand over here. Well, thanks for not leaving after the break. I was a little worried taking the last session that we were going to have a half-empty room, but again, thanks for staying. I'm going to talk a lot about implementation, really questions that you need to answer if you're going to think about doing implementation. And I'm going to briefly give you some examples of how to answer those questions, but by no means is this a playbook of this is how you're going to do it. There are many ways to solve the implementation problem, and it's probably going to be dependent on your health system, but hopefully you'll have some really key questions to address when you get back to your institution. Here are my disclosures. And so objectives, we're going to talk about components of the program, strategic questions, so high-level questions that you need to answer, and then really tactical questions. How do you bring this to the patient, and how do you bring this to patient care? So where do you guys think we are in this journey? Who thinks we're at the very beginning of implementing pharmacogenomics in clinical practice? So maybe those first two question marks. Raise your hand if you think we're there. We're trying to answer that can question. All right, so we got a lot of hands there. What about people in the middle who think, you know what, maybe we can do it, but we're trying to figure out what are the best practices of doing it? Who thinks we're there? We get a few more. And who thinks it's actually routine care and we don't need to be here at this conference? All right, nobody answered that one, so good. So can we do this? Well, I'm going to say we can, and here are a sample of papers published in the last year about how you can do it. So it's about 25 different papers that talked about different tools or processes for implementing pharmacogenomics. So I think there you can do it. And then you guys have heard about many of these tools already today that help you do it. So those of you who answered that question, we're at the point of understanding, can you do it? I think we really need to recognize that we're past that point. The answer is you can do it. Now the question is, how do you do it, and what is the best practice to do it? And I'm going to share with you how we address that at NorthShore. So when we think about NorthShore, anybody know NorthShore before they came here? We got 1, 2, 3. So I'll clarify, this is NorthShore in Illinois, not NorthShore in Long Island. That gets confusing sometimes. But NorthShore University Health System is really a community health system. It's the type of health system where most of America gets their care. It's not a large academic center. It's got 4 hospitals, 800 beds, so it's a reasonably sized hospital. We have over 100 physician offices. We have about 100,000 ED visits in any given year. And most important to pharmacogenomics, we have an integrated outpatient and inpatient EHR. So if I get the results in one place, I can use it everywhere else. And that is incredibly key to realize the full value of pharmacogenomics and really impacting the patient's experience throughout their care process and not just in one encounter. So strategic questions to ask before you even begin doing pharmacogenomics. Why are we doing this? Where does it fit into our program? And who's available to help us do this? You're going to really want to answer these questions because that's going to change how and what you're trying to do and what goals you're going to try to go for and how you measure the value of pharmacogenomics in your institution. And that change in value measure really will drive what are your first implementations, your second implementations, and what does that home run look like in your particular institution. So a lot of times when I ask people, why are we doing this? They give me an answer similar to what you see on the screen. We're going to do it because we have therapeutic alternatives to choose from, our clinicians have this problem, and we're going to give them more data to do that. And if we do it correctly, we're going to get safer, more effective drug therapy. We're going to get increased adherence, decreased hospitalizations, decreased healthcare costs. Those are all great answers for why you want to do pharmacogenomics, but that's not the point of this question. These answers tell you why pharmacists should be involved, because those are all things pharmacists have been charged with over time. But the real question about why is your health system or institution going to invest in this? Because it's going to take time, it's going to take money. You're not going to be able to do it from the first day. And if you can't define this answer, I'm going to caution you to go down this path because you're going to be throwing your effort into a black hole, and it's not going to go very well. All right, so NorthShore's answers to why. Well, we wanted to meet the pharmacogenomic needs of our patients and providers. And so you're like, well, how do patients have pharmacogenomic needs? Well, we happen to live in an era where a couple of things are happening. We have consumer-driven or patient-driven healthcare. We also have genomics becoming in the lay media and lay public. So I'll give you 2 examples there. One is the Mother's Day card I bought last month at Target. It had a double helix on it. And the message was, thanks for the excellent genes, Mom. Happy Mother's Day. So this is a card that I bought at Target, and it had DNA on it, so I had to buy it, right? And my mom was really proud about it. But anybody else could have bought that card, right? And the fact that they made that in mass production really states what our relationship with DNA has become in America. The second thing I'm going to bring up is a TV show that I watch. Billions. Anybody watch that show? It's on Showtime. And we got a couple hands about it. 2 episodes ago, there was an investment strategy, and the company they were investing in was a pharmacogenomics company. They didn't use the word pharmacogenomics, so they said, we're going to use genetic information to determine drug therapy. And the company got evaluated at about $4 billion. So they believe it has a lot of promise, and I hope you guys do too. So patients have pharmacogenomic needs, and they're going to probably go out and interact with genomic data, and you need to have a system to support that, or at least we felt that way at NorthShore. We also wanted to provide reliable and consistent interpretations of the data. This is a huge challenge for a health system that uses multiple testing labs. You may get a CYP2D6 answer from one lab and another one from another lab, and one is a normal metabolizer, one's an intermediate, but when you look at the genotype, it's exactly the same. Kelly, I hope you can solve that problem in the next couple of weeks. I'd really appreciate it. We also wanted to use pharmacogenomics as the tip of the iceberg to get genomics into care at NorthShore. We thought this is something easy. Everybody knows 2 people who've had the same medication and got different responses. And so it made sense to our clinicians, It didn't have a lot of ethical concerns, and so we wanted to use this as our learning case for how we were going to do genomic medicine moving forward, because we're going to be faced with that, and we had to deal with it today. And then also importantly, we wanted to differentiate ourselves from everybody else in our marketplace. If we thought we could do pharmacogenomics and precision medicine, that could drive patient care to us. That could bring in new patients, it could retain patients. It's a really important idea because that gave me support from the top down to be able to do this and use these resources. And our mission statement for our pharmacogenomics program became using and providing next generation healthcare through prudent and responsible applications of pharmacogenomics data. Where does it fit? I told you that pharmacogenomics was the tip of the iceberg, so it fits in this much larger structure of personalized medicine. But at our institution, what we decided was, for all of the great work we've heard about today in the research realm, let's see what happens when we do it in routine care and drive it to patient care. So at NorthShore, research is a very small piece of it, and 99% of the genetics that's done is just through patient care and routine patient care. We have testing, we have clinics, we have systematic integration, we have education. If you want to talk to me more about that later, happy to do that. So who's involved with our program? We identified some key players. So you have stakeholders, administrators, clinical leadership, providers, and patients. So this gives you a top-down support, a bottom-up support, and a middle-out support. And then everybody can begin to get on the same page, and you can begin to move this boulder that is personalized medicine. And then personnel, who's going to do the work? We have a program leader, that was myself. We have physician champions. We do testing, so we have laboratory specialists. We've got to translate that information, so informaticians are important, educators, and then patients also become very, very important. They need to participate in personalized medicine if you're going to derive the real value from it. So I want you to think about the slide for a minute. The only group that is in both of these slides are patients. This needs to be patient-focused and patient-driven if we're really going to maximize the value of genomic care. So if you answered those questions, you have an idea what's going on. Now let's think, what are the tactical things that we have to do? We have to obtain genomic information, we have to interpret it, and then we have to return those results to patients and providers. To do that, you need to do probably 5 different things. You've got to generate genomic data, so that's genetic testing. You can do that in-house, you can do that out-of-house, it just depends. You've got to take those genetic results and aggregate them and standardize them. We do that through a genetic results database, all electronic, happens automatically. We have a knowledge base that's driven, that takes those variants and tells us what they mean and gives us interruptive alerts. And then we have a user interface, because we've got to deliver to that somebody. I didn't say an EHR, I said a user interface. Why? Because sometimes it's the patient that needs that data, sometimes it's the provider, sometimes it's 50 providers, right? Sometimes it's family members. It just depends on the use case, and you need to come up with a solution that meets all of those. So sometimes I tell you that PDFs are really, really bad, and I think they're really, really bad when you're trying to do interruptive alerts, because you don't have discrete data. But when I'm trying to educate a patient, PDFs actually might be a really nice way to handle giving them that information. So think about that solution, and then education becomes incredibly important. I told you we do most of this automated. That's where we are 4 years into the process. On day 1, it was manual. What was it? It was me looking at the report, typing the medical note, and moving on with our day. So you don't have to hit a home run to begin with, and don't expect that you can. So we talk about needing discrete results, but you can start out without doing discrete results and still get somewhere. So let's look at obtaining genomic data. One of the really important questions comes down to, when do you want to do it? Preemptive versus reactive. For a long time, we thought about this as one or the other. Probably turns out they're more likely to be on a spectrum, that you go from a preemptive approach all the way to reactive, and your value proposition for each of those points is going to change. So if we look over here on the preemptive side, these are the patients that get me really, really excited. A healthy patient, no medications, gets tested on a multi-gene panel. So when we do that, the genomic data has the ability to affect every medication decision thereafter, because we don't have prior experience about it, right? So that is the most value in the way in which most people think about the value of pharmacogenomics today. Can I make a change to somebody's medications off of this without having to think about some additional factor? And I would love to test everybody at NorthShore in this capacity, but that's probably not realistic. Maybe somewhere it's a little more reactive. You have a patient who's starting antidepressant therapy and getting a multi-gene panel. So here you've narrowed it down. You're reactive because you have a particular indication for why you're doing it, but you're preemptive in the nature of you have all of these other genomic data available for you for future things. So I'm going to immediately use this information for antidepressants, and then I have stored up value thereafter if I can deliver that in the system. Then you have your traditional reactive test where you're going to test HLA-B5701 because of abacavir. One test, one problem, we're done, we're moving on. Interesting value, very clear, really pointing in the time of care, easiest thing to get paid for. You'll get that paid, no problem. Then we have the most reactive, and this is somebody that's failed every known therapy for a given condition. Why am I going to do pharmacogenomics in them? I'm not going to help them choose between therapeutic equals. Is there value there? Well, our clinic experience says there is, because patients begin to say, you're able to explain some of my journey along the way, and they can begin to think, it wasn't something that they did wrong about why they failed all those therapies. It's something that is embedded in them that they couldn't have overcome. And that can be incredibly and powerful and engaging for patients. We look at Jillian's case from earlier today, right? That patient who the results didn't help them at all for which treatment plan they're going to do, but the treatment plan they developed without pharmacogenomics, they felt more comfortable in. We cannot ignore this value when we're thinking about a patient-centric approach. Interpreting data, you've got to take something that looks like this, turn it into something like this. CPIC is a great place to get started, but you're probably going to want to tailor that to exactly what your clinicians need. This is probably 20 words. Our experience in NorthShore says 20 words is too much. You need to get it down to 3, and if you can give it to me in a picture, I'll be much happier. It's kind of the feedback that we're giving. In the care setting, right, like when the patient's right in front of you, you want the shortest answer with the most impact. But in another setting, this might be helpful, or even a longer answer. I know Dr. Johnson talked about how nobody clicked on their more information button. We'll see where that plays out as we move forward in the future and people get more comfortable with genetics. Returning the data. So for returning the data, education becomes incredibly important. And there's educational needs about clinical knowledge, what is pharmacogenomics, what are the drug gene pairs, what are the testing interpretations, the legal issues associated with it. But actually one of the most important things is operational knowledge. So you can build all of the tools in the world, come up with all the resources that provide all that education, but if you don't tell anybody how to get to it, it's never going to get used. If you don't tell somebody how to use the interruptive alert that you just provided for them, it's a waste of everybody's time. So don't forget about operational knowledge and how are you going to educate that. And you can use that operational knowledge and those tools to then drive the rest of your education agenda. So what does pharmacogenomics look like at NorthShore? Here's our timeline, pre-2014, before I got there. We have no idea what happened. I think it was a little bit of the Wild Wild West. If you wanted to do it, you could do it. There were no rules, no laws. You could do whatever you wanted. Probably gunfights out on the lawn in the evening. I don't know. In August, we started a pharmacogenomics subcommittee of 2014, so this is similar to what Dave described. It reports through an oversight structure similar to what St. Jude has, it's similar to what Mission has and what Florida. This thing seems to be recreated over and over and over again as people do implementation. We created 3 drug gene pairs in August, or in January. Then we started our clinic in March, and I'll talk about that in 1 second. Then we moved to a more robust and a third-party solution for our clinical decision support, because it takes a long time to build these things, and so we wanted to leverage somebody else's expertise. In October, we started in-house testing, and then in January 2017, we began to allow patients to do the testing in their house, deliver education via a video, and we began to meet the patients at their care site instead of making them come to us to get pharmacogenomics. So we really were taking the healthcare to the patient and became incredibly important. So 2 delivery method. We're going to talk about the clinic and the at-home testing first. So the pharmacogenomics clinic. It was designed as 2 different visits. The first visit was really talking to you about, here's what pharmacogenomics can do, here's what it can't do. In your particular setting, this is what we think we might be able to do. Here's the cost, $225. Do you want to do it? Let the patient make a value-based decision. But it was about giving patients access to this and aware of all of the information so that they could make their own value-based decision, and we weren't telling them whether they should do it or not. Once we knew that, we then would see them again a couple weeks later when we had the results and go over what do the results mean, both for their current care and their future care. Both of these were visits that we had with a nurse practitioner, so we were dropping office visit charges for this, and this was revenue generating for our institution. The testing was not. We didn't want that to be the case, but we allowed the clinic visits to do it. That cognitive knowledge, because some of these patients will come back in a couple of years and want another clinic visit. We don't need to retest them, but we can tell them more about their interpretation. So it wasn't just a one-time thing. For the at-home testing, any provider at NorthShore can order it for you. You go home and you watch a 4-minute video. That is that 1-hour clinic visit distilled down to 4 minutes. It's a little depressing that I could actually do that, but we were able to do it. And then you get a kit sent to your house. You send it back to the lab, and we deliver the results in the EHR. What does that look like? Well, we took 2 different approaches here. For the patient, we wanted to deliver a drug-focused approach to begin with, because that's the unit that they're most interested in. So it was really, really important to do that. But because one of our goals was to use pharmacogenomics as the beginning of the argument for why you need to do genomics, we didn't want to forget about the genetics, right? We wanted to use this as a way to get them familiar with genomic concepts, so we included a gene-centric approach as well. And so you get both, and patients can interact to their comfort level. But it was important to recognize we needed to deliver that second piece to deliver on the why we were doing it for our institution. For our providers, we again wanted to take a drug-centric approach. And so there's this profile that's available, and this is an alphabetized list of all of the medications that are a problem that they can go to at any time for a patient who has results. This might not even be the best way to do it. It might not be the most valuable piece of currency for clinicians. They may not be thinking about, I'm going to treat this patient with this drug. They instead might be thinking, this patient has this disease, what drugs could I treat them with? And maybe delivering it in a disease-centric approach may be better. We're working on what that might look like. And of course, as everybody else has, we have interruptive alerts that have distilled messages with actions that follow up when you have life-threatening or severe toxicities associated with the pharmacogenomic decision. So we delivered the clinic and the at-home testing, and we thought, well, we're doing 2 different things. One is an hour-long visit, one's a 4-minute visit. It's kind of, we don't know, one's really controlled and one's a little loosey-goosey. Can we compare those, and are there differences? And so we did this patient study, and we wanted to assess patients' perceptions and utilizations of pharmacogenomic testing, and we wanted to determine if the delivery method was important. I'm going to tell you there was no difference between the 2 delivery methods. Everything we asked for, there was no difference between those 2 groups. So then we began to aggregate the data to say, well, what were patients doing with the data? How were they feeling about it? I just want to share these 2 results with you. So the first one is, I felt more confident in my medications when I had pharmacogenomics available. And you can see that over 70% of patients either strongly agreed or agreed with that statement. So we're getting to a place where we're making patients more confident in their treatments, and if patients are more confident in their treatments, they're more likely to work. And what this allows us to do is highlight what Jillian highlighted, which is pharmacogenomics is going to be important for everybody, regardless if you have a pharmacogenomic variant or not. If I can make you more confident by telling you that the medication doesn't have a genetic problem, that's wonderful to me. And as you can imagine, if we make you more confident in your medications, you're more likely to take them, and we're beginning to see this signal here. I'm not saying this is definitive proof of the real value of pharmacogenomics, but it's definitely a signal that we need to figure out how do we do more research on this and prove that this is actually a value case for pharmacogenomics. I'll show you this quote. I'll let you read it for a second. If this is the only reason why we do pharmacogenomics, I am plenty happy doing it. If all it does is start a conversation so that physicians and other clinicians begin to listen to their patients, wonderful use of $225 as far as I'm concerned, and I think this patient would agree. And so I want to show you how we changed who is utilizing pharmacogenomic services by going to a more open approach, one where we're giving patients more access to the data, where we're meeting them at their place as opposed to making them come into the clinic. In 2015, it was mostly psychiatry doing the ordering, a very targeted patient population. At the end of 2017, primary care was the leading orderer of pharmacogenomic services. So what this means is we're moving closer to that preemptive model where we're getting healthy individuals or individuals that haven't made it to a specialist first, and we're beginning to impact their care there. So this makes me really excited. So it makes me wonder, maybe we're closer to the end than we think, but we're only going to get there if all of us begin to work and figure out these are the best practices for pharmacogenomics. So please join me on the journey. Happy to help you, and I'll take some questions.
Thank you, Mark. We have time for a couple of questions. Anybody care to step up to the mic and ask away? This is kind of the culmination of a lot of what we talked about today, and it's something that I think is important to represent as, yes, in a way, one size doesn't fit all. Every institution is going to be different. I'm kind of curious about, oh, I see a question. Go ahead, Brian.
It's impressive, the increase you got in primary care. I assume that must have meant you've had a significant preemptive education.
Yes. So I showed you that there were 100 medical group sites. I've been to 100 medical group sites. So I went to every single one of them, and I sat there for 4 hours in their office, checked my email, but was there to answer any questions that they had, gave a short presentation. That was incredibly important because they needed to feel like they had a support network that they could go to. So when they had a question and they wanted to email somebody, there was a face to that email. That phone call, there was a face to it. So education, huge component, going to have to drive it, going to spend a lot of resources doing that.
Go ahead, Zach.
Zach Rivers from here at the university. So at an academic health center like we are, sort of compared to a community health center where you are, what are the steps that researchers here should be taking, in your opinion, to support the dissemination of this into the community and sort of out of academia?
So I think one of the really important things that we need to think about with implementation is how do we roll it out in an implementation science framework so that we can replicate it and we can do more than just have some guy up here from a community hospital tell you what he did, and instead you actually have real data that's structured and has the scientific rigor behind it to say this is really the best way to do this. And I hope in a couple of years you don't have me doing this anymore. You have a lot more studies with graphs and pie charts and those types of things.
Thank you.
Mm-hmm.
So one last question. I just wanted to ask if you might be able to describe, of all the steps that you engaged in, which were the most challenging to overcome in your setting, obviously, but things that maybe might give a hint to some that emulate that kind of a model elsewhere.
So one of the biggest challenges was understanding why does my institution want to do it? Seems kind of odd, right? So when I went to NorthShore for my interview, they wanted to hire me as a research scientist. And I don't do research, right? I'm a clinician, I'm a PharmD by training. And we had to figure out what is the need that NorthShore has defined? Because if I understand why we need it, then I can come up with programs that meet that need. Otherwise, if I didn't know that answer, and it was a lot of different answers to a number of different people, so it was really identifying the threads, pulling them, and sewing it together into a story about here's why we need to do it. So I had to be a little bit of a salesman, but I had to know that to begin to say, this is the first project that we went after, so we could get an early win that everybody agreed was an early win, because it's a long journey, takes a lot of resources, and if you ask for a bunch of time and money, you got to have something early, otherwise people lose interest and funding cycles run out and all of those types of things.
Well, in spite of you not being a researcher, I'm still impressed that you publish in the area and on your experience, and that's helpful to others.
So Mark, what are you guys doing in terms of measuring value? Are you measuring costs or outcomes in some way?
Yeah, so as far as the clinical value of it, because our testing approach doesn't limit to any targeted segment, It's really hard for us to show you that it's really helpful in depression or it's really helpful in pain management. It's more about an overall approach for the healthcare system. So our patient study begins to have some value, and I'm really struggling with, we kind of have this last mile problem. I can generate a whole bunch of data, I can interpret it, but can I deliver it in a valuable way? It's kind of like I'm Amazon and I have a warehouse, and now I got to figure out how to get it to my patient in 2 hours like they can anywhere in Chicago. That's the problem that I'm solving, and how do I do that in a way that changes patient behaviors? Because that's where we'll see the value, and once I feel like I confidently can do that, then I'm happy to go measure the values of pharmacogenomics. But I'm struggling with exactly what that looks like, so I hope some of you guys begin to tackle that problem and can tell me what the answer is.
Thank you once again, Mark, on behalf of the group. I appreciate your insights.
Thank you.